Let's talk about bacteria. You know what bacteria are, right? The oldest and smallest organisms on this earth. These little microbes are really, really small, but they perform some huge tasks, such as bioluminescence, virulence, sporulation, and biofilm formation. How do these bacteria perform such tasks? Well, the answer is group work. No, not the type of group work that you do in school where you're the one who does all the work. No, this is much different. To perform such big tasks, bacteria have to reach a certain quorum, and the way they communicate this with each other is by using quorum sensing. On the surface, quorum sensing is quite simple. Bacterial cells release molecules called autoinducers. When in an environment with a low cell density, these autoinducers just diffuse. The real magic happens in a high cell density environment. When there are a certain number of cells, enough autoinducers are produced to be detected by receptors in the cell cytoplasm. Once these receptors detect enough autoinducers, they spark the gene expression process. Simple, right? But when you think about it, there are 2 billion different species of bacteria, and many of them have their own molecules and sensors. This makes it a bit more complex. The good thing is that there's one big classification of bacteria that comes into play: gram-negative and gram-positive. Quorum sensing in gram-positive bacteria is kind of like sending a message to a medieval king. The message is sent out, and once it reaches the castle's outer wall, it has to be inspected and taken to the king by a messenger. Just like your message has to be actively transported through the castle's thick walls, the autoinducer has to be actively transported through the thick cell wall of a gram-positive bacteria. The autoinducer in gram-positive bacteria is usually peptide, and it activates kinase activity in the cell. This causes phosphate to be produced and passed to a response regulator, which activates the transcription of genes. In gram-negative bacteria, it is more like sending an email. The message can be passed to the recipient much faster and without as many checkpoints. So, gram-negative bacteria have acyl homoserine lactone or AHL autoinducers that easily diffuse through the thin cell wall. AHL binds to one side of a LUXR-type transcriptional regulator protein, and the other side of the LUXR protein binds to DNA. After this, RNA polymerase arrives and begins the gene expression process. Now, how does knowing this help us? Well, as you know, there's many pathogenic bacteria, and our usual way of killing them is antibiotics. But in this modern world, many of these bacteria are now resistant to antibiotics. Now there's only one way to stop them, is to stop their communication so that they can't express any more genes. The only way to do this is to block their quorum sensing, and this is why I think that quorum sensing is going to be a very important topic to learn over the next few years so that people can figure out how to stop quorum sensing in dangerous bacteria and help out people with diseases.